good afternoon to you all. Lady Brabon College, Department of Botany is organizing a one-day webinar, Recent Advances in Biology. I welcome all our participants and our speakers. Our first speaker today is Dr. Partha Chakrabarty. He is the Principal Scientist, Cell Biology and Physiology, CSIR, IICB. Dr. Chakrabarty's research interests include molecular basis of different metabolic syndromes like type 2 diabetes, non-alcoholic fatty liver diseases, obesity, and certain types of cancers. Today, he will be speaking about experimental approach to biology. So, I again welcome Dr. Partha Chakrabarty and you can start your talk. I would request all the participants to mute all your audio and videos while this deliberation is on. So, welcome all to the webinar.
So as you know, so first of all, thanks to the organizers and particularly Vidisha for this introduction. And as you can understand that I'm no botanist and I'm presenting in a botany department, which is what I see as step anyways, but what in the experience, what I gathered in last decade or so, based on biology teaching and experimentation and the research. So I just like to share that with you all. And this presentation is primarily focused on looking at the students, particularly the undergrad and postgraduate students. So what I do today, I'll not burden you with a lot of information, but rather I'll talk about certain concepts and how to approach and how to study biology from an experimental point of view. So experiments will be one of the focus area, and how we can approach biology teaching as well as research in terms of experiments, that I'll be talking about. So let me begin with this this slide. So when we teach or when we study biology, we study a lot of books, right? And you can see some of the books may be familiar to you, but for students, what most of the students uses for their study material is not the books, but the class notes, right? That is shown in the middle. So we take notes what our teachers tell us, and we make a note out of it, and we try to answer the questions given in the exams, right? And so this is excuse me. So this is the approach, right? So a student who studies well, so to him or her, the things they are well aware of it and they understand them, right? And from that material, questions are given in the exam, right? And the students give answer, right? And if you answer right, so you get a good grade. And we say, oh, you are a brilliant student, right? And that's how the pedagogy works in the field of biology and most of the sciences, okay? So what I would like to look at, to emphasize the green box on the left, okay? So the things which we are aware and we also understand. So there are two qualities, awareness and understanding. So knowing, mostly knowing the subject, okay, the material, right? But if we expand this idea into four categories in making a matrix, and you can see this green zone, orange zone, and red zone, which we are kind of familiar with, right? And if we make this matrix in terms of known and unknown, so the green box, which I have iterated before, you can see the things we are aware and we understand, right? Now the orange zone is somewhat a difficult place where you have, so there are things which are aware but we don't understand, right? And the red zone, which is a really red, where neither we are aware nor we understand the subject. But on the left bottom corner, if you see, so there are also things that we understand but we are not aware of. So these sounds really too philosophical and too complex for you. but I have given an example of a green zone, which you all know what is taught to us and what we write in the exam. So we'll come back to the orange zone in the next slide, but before that, on the left bottom corner, so you think, so things we understand and we are not aware of. What kind of things we do? So this is a area of cognitive neuroscience, for example, where we call it a domain of unconscious, for example. So we understand a lot of things that we should not tell lie, we should not cheat in exam, and we should not make bad comments to our peers or parents and friends and families, but we do that, right? So we understand that, but when we commit these mistakes, we are not aware when we are committing, right? Okay, so let's go to the green zone. So it sounds weird, right? That so things we know, we are aware of, but we actually don't understand, right? Now let's see an example, and it will be clear. So this cartoon, I hope you all are familiar with this cartoon now. And so we now quite aware of coronavirus, but still we don't understand, right? So to make coronavirus understandable, what we need to do is to conduct some experiments to make things understandable, comprehensible, right? That's where the domain of experiments comes in. And from that, we have to design experiment on coronavirus so that we can make better understanding. So now when we talk about experiments, we have all done it in our even high schools, and we make, particularly in chemistry, if you remember, so we make 3 columns, right? We describe experiments, then we write down observations, and then we write inference. So these are pretty straightforward 3 steps model, where experiments is described and you observe something and you conclude something, right? But for biology, it's not that simple. And that's why in the history of science, biological experiments came very late, and still biological experiments are being taught and new biological experiments are conducted. Now, why is the problem? The problem in physics and chemistry is more of a deterministic in nature. That means where your input actually controls the output. So it depends on how much force you give, you know what will be the speed of the object. So in biology, the main problem, as you see, the output is independent of input, right? So for example, we always experience this, and I'll come to that problem in the very next slide, that for example, if we ask about what is the age of students in a class, there is no single data point, right? So we always see a distribution of age or height or the basic parameters, and so we need to make some statistical maneuver to come up with a data point, right? So that's why, since biology is always probabilistic, and it's not the entire population of a species or an organism or cells or molecules, they behave similarly in a deterministic fashion. So designing experiments become really difficult in biology. And you can see, again, if you go back to corona, now you see, so the questions like, so who will be infected? When will be infected? How will be infected? How much virus is needed for clinical symptoms to occur? And finally, why me? I'm getting infected, or others are infected, right? So all these questions, and none of them, as you can understand, if you look at the media, news media, or a digital media, you would see there are a whole bunch of versions are available, and you see a whole lot of confusions arising, right? And also, if you think about the therapy, so whether a drug is available and how to make a drug available, whether a vaccine is there, what about plasma therapy, what about traditional medicine, there are a whole bunch of questions, and nobody knows which way to go. But for example, if you think about a bacterial infection, say for example, cholera or salmonella, typhoid, so everything we know. So because the experiments on salmonella or cholera is already done, and we have a specific recommendations and specific information on that, and for corona we don't, right? So there lies the confusion. And to get out of these confusions, we need to conduct experiments. And so I'll just give you a few examples to our students, and both are related to one biological event we call phosphorylation. Now, so we all know about this. I guess that you all know about glycolysis, right? So whenever glucose enters the cell, it is first converted to glucose 6-phosphate, and which again by multiple steps end up in production of 2 molecules of pyruvate. Now, so just think about, so we study these, we memorize this, we write it in exam, right? But just think about the very first step. So glucose to glucose 6-phosphate, right? And also, we also know that glucose 6-phosphate can be converted to glucose by an enzyme called glucose 6-phosphatase. Okay, so now the question is, so we know this, it's written in the textbook. Now the question is, how do we know that phosphate group is linked to the 6th carbon, specifically of glucose molecule, right? And how do you know that a particular enzyme, glucose 6-phosphatase, removes that phosphate only and not the other phosphate from the other residues, right? So that kind of questions we usually never ask when we study biology. And I would emphasize that our students should try to think about these issues whenever we read about a statement or a fact, a biological fact. We should be able to think about the experiments required to establish this fact, right? And these facts actually came out of some experiments. Without experimental proof, science does not become facts, right? So scientific facts comes out of experimental data. And so for just to think that a glucose 6-phosphate becomes glucose by a process called dephosphorylation, what would be your experiment? And I would insist our students that you should think about it. Similarly, I'll give you, so this is a phosphorylation of a metabolite. Now think about a phosphorylation of a protein, right? So things, again, these things we are aware of and we also understand. But what we don't know is how these facts has arisen into our textbook, how they come in, right? So if we study signaling pathways anywhere, whether it's a plant world or an animal world, we'll see proteins are often get phosphorylated, right? So phosphorylated is mediated by kinases, and the opposite one is dephosphorylation, which is mediated by phosphatase, another set of enzymes, right? So what would be the experiment to show that a particular residue, a particular amino acid of a protein gets phosphorylated or dephosphorylated, right? We often read these signaling pathways, but oftentimes we don't really pay attention to how we came to know that this particular protein gets phosphorylation on a particular cellular or organismal context, right? So that's where, so I request and emphasize to our students that you should try to think about experiments and try to understand methods in biology where we can actually generate biological facts which are often written in the textbooks. Okay. So now, as you know that, for example, in exams, you are asked some questions and you have to answer from what you know already, right? But when you are designing experiments, the method is a little bit different. And it's a bit complex, but I'll just describe it to you briefly. So here in experiments, first you have to ask a question. And based on that, you have to come up with an hypothesis, right? So what could be the possible reason for that particular question? For example, why corona is being prevalent now? now we have to think about an hypothesis, right? And from hypothesis, you have to design an experiment, and you'll get experimental data. And these data may conform with your hypothesis, or then you accept it, and you ask the next question. Okay, so when you know that corona is transmitted by droplet infection, for example, then you ask how to. The next question could be, what would be the methods to stop droplet infection, for example. But if your data suggests against the hypothesis, against your hypothesis, then you reject it, and again you build up a different hypothesis, a new hypothesis. And that's how science progresses over time. But in biology, as you know, as I have already said, that it's a probabilistic in nature, the entire system. So the experiments are a bit different from traditional chemistry or physics experiments. So for biological experiment, you always need at least two groups to start with, right? One is called control and one is test, right? So the control, for example, so this is the group which does not include the thing being tested or is used as a benchmark to measure the results of the other group, right? So you know the control, what is going to happen. So you always have to design an experimental arm where you will definitely know that the results will be either positive or negative. So there are positive controls and there are negative controls, right? And then you do a experiment, separate experiment. So now here biological experiment differs from chemistry experiment. You have not just three columns, but you have two groups, control and test, and under each group you have three columns. Okay. It sounds a bit complicated, but let me give you a high school experimental evidence and you will know this, very simple. So, excuse me. So suppose you have two identical pots, you put some 10 bean seeds and you water them in one and you don't, and you see the plants are coming out of this pot, and 9 out of 10, whereas 0 out of 10, right? Again, you see it's not 100%. Even in this example, you have 9 out of 10. But now this experiment will serve as a positive control for your next experiment. Suppose you want to study impact of certain toxin or an impact of a certain fertilizer on a plant growth, and then you can use this as a control group. So you know this many seeds, this spot, this is the soil amount, and this much of water is to be given to have at least 9 plants to grow, right? So that's how we generate controls, and which can be utilized for the future experiment. Now, just to go into little deep into this technique, just think about, so the experiments has to be done in a very controlled fashion. So what do you mean? So the control is, as I mentioned, so the controlled environment is critical. That's why we need laboratories where you can control most of the, or a greenhouse, for example, in the plant science. So you can control a lot of other variables, because the entire biological system, as I have mentioned already, it's very probabilistic and distributive in nature. So that has to be controlled. Otherwise, we won't be able to elicit any response from any intervention. And intervention is basically our experiment, right? So intervention, we call it independent variable, and what we observe, what we get is a dependent variable, and entire system has to be controlled in nature. And I'll just give you two examples from cell biology, molecular biology. So one is, for example, is Western blot, where we study proteins, and we can have a lot of questions, for example, to see whether a particular drug or a chemical, how it works on a signaling pathway. There are tests for conformity tests for HIV, and there are a lot of other areas of serum, tissue, and cells where we study proteins. And for that, for example, we use Western blot, right? So suppose we want to see whether a particular type of hemoglobin is present in a particular, so for example, in thalassemia. So we can do electrophoresis and Western blot, so for example. For example, agarose gel electrophoresis, another example which is very commonly used in biology labs, where we primarily detect our DNA and DNA or RNA samples, and for PCR products and other clones, right? So for example, so nucleic acids, we mostly depend on agarose gel electrophoresis, whereas for proteins, we mostly depend on Western blot analysis. And for biology, as I already mentioned, so because we cannot control the variations in organisms in the wild, so we need to sort out, we have to go to few model organisms where we can actually control, for example, their genetics. Okay, so the laboratory animals are there. Drosophila, one of the classic example, and C. elegans at the bottom you can see here. The mouse, where you have the homogenized on the homogenized genomic background. This gangetic fish, you might have heard about zebrafish, and in plant science, Arabidopsis thaliana is very commonly used across the globe, right? Where we can do actually conduct experiments in a very controlled fashion. So, but the question is, the big question would be, so whether we can perform human experiments, because humans are also subject to biological variation, and we want to answer a lot of questions in human biology, right? But can you do this? And that has been a big debate over decades, and there are a lot of ethical issues involved. And some of you may identify this picture. She was the first volunteer for the coronavirus vaccine, for example. And this is an human experiment is being conducted. Okay. But human experiments are being conducted over 100 years now, and there are a whole bunch of really dirty and horrendous experiments have been done. We know about the Jews with Germany under Hitler's regime. A lot of human experiments were done. People were subjected to poisonous gas and etc. And this is one of the dark history in, for example, in modern medicine. These experiments was conducted in the United States on the black males. As you can see, the objective of the study was to see what is the clinical course of syphilis over time. And you can see it's not that bad. So this is one of the dark example in the history of mankind, that where these black males were injected with syphilis organism, and they were followed up to look at how do they behave over decades. And that was human experiments, no antibiotics, nothing was given to them, and they were left to die. Okay, so these kind of experiments are now banned, and these horrendous experiments are now banned. And now what is mostly used is called randomized controlled trial. So again, you can see it's controlled, right? Now, problem is, how can you control? Even we cannot control students in a small class in this Zoom meeting event, and then controlling and looking at, say, for example, effect of a drug is really, really problematic. So what is done, you can see this is a simplified design of RCTs. So you take a whole bunch of patients, you statistically differentiate them in two groups, control and treatment. So for example, treatment groups are given medicine, controls are patients, control group is also given a medicine, but that's a dummy medicine. It's called placebo. And then they're followed up for clinical response. And as you can see over here in the treatment group, you see there are people less having complication, whereas in the control group you have more complication. So, and there are statistical ways of comparing these results, whereby you can conclude that, okay, this treatment has some impact or a better impact than a placebo, right? So that's how human trials are conducted these days. And so So I would like to stop here. And so this is my group, the research group. And thank you all for listening. And I'll be happy to take questions now.
Welcome back to the webinar. Our next speaker is Professor Amal Kumar Mondal of the Department of Botany and Forestry, Vidyasagar University. Professor Mondal's research interests include chemotaxonomy, molecular taxonomy and palynochemistry, ethnobotany, biodiversity and conservation. Today, he will be talking about a molecular method for identification of the medicinal plants based on DNA barcoding. So, Professor Mondal, please take over.
Good afternoon, everybody. And first of all, I would like to thank the principal of Lady Babon College, Professor Sivli Sarkar, and the organizers of the webinar, that is the Postgraduate Department of Botany, Lady Babon College, for giving me this opportunity to talk on the topic, a molecular method for identification of medicinal plants based on DNA barcoding. So the traditional system of medicine implements medicinal plants to cure various diseases. Out of 7,500 wild plant species used by tribals for traditional purposes, about 950 are found to be new claims and worthy of scientific scrutiny. The traditional herbal medicines have gained popularity worldwide, and about 80% of the world population utilizes traditional medicines for well-being and healthcare. The world market of herbal medicines, like herbal products and raw materials, has been evaluated to have an annual growth rate between 5% to 15%. India has a great treasure of traditional knowledge. We know that India has a great diversity in the plant and as well as in animal, and India also called a mega diversity center. Not only in the vegetational point of view, on the medicinal plants point of view. The demand of medicinal plant related trade in India is predicted at rupees 5,000 crore per annum. A total of 355 crores are invested in international market per annum for Ayurvedic preparations. First of all, my reason for selecting this topic. An important part of the plant inventory of Indian herbalists consists of barks and roots, which typically possesses few physical characteristics that enable accurate morphology-based identification. Herbalists are able to provide information about the local name of a plant product, its medicinal uses, and their origin. But this information may be imprecise or insufficient for species identification purposes, especially considering that herbalists often do not possess knowledge of medicinal plants in the wild. Some medicinal plant products have multiple synonymous names, and in other cases, the same vernacular name is applied to multiple plant species. Thus, the identity of a plant sample, which may be bark, root or seed, etc., brought from this herbalist has so far presented a challenge. In addition, since the collection of roots usually requires the whole plant to be dug up, the trade of medicinal roots has a large impact on natural plant population. For example, thus the identity of the plant being sold in these markets has conservational as well as medicinal implication. For example, rare or endangered species could involuntarily be collected if they are easily confused with their more abundant relatives. Likewise, increasing demands for medicinal products may lead to local over-harvesting extinction of otherwise non-threatened plant species. The traditional method for identification of dried medicinal plants and plant parts is by various senses like taste, sight, smell, and touch. And now next, the problem is the adulteration. The adulteration of medicinal herbs with closely related species is the main cause of concern for the herbal industries today. The reason for this might be misidentification or scarcity of correct herbs. The types of adulteration. First of all, substitution with inferior commercial varieties. Number two, adulteration by artificially manufactured substitutes. Number three, substitution by exhausted drugs. Number four, substitution by superficially similar but cheaper natural substances. Number five, adulteration by addition of worthless heavy material. Number six, addition of synthetic principles. And number seven, use of vegetative matter from the same plant. What are the fate of adulteration? The adulterated drug in some cases may be mortal if substituted with toxic adulterants and become life-threatening. Hence, the correct formulation is important for the herbal drug to be effective. Now, what should be our objective? The first appropriate measure can be taken if a reliable method for species identification of medicinal plant products existed. Number two, the latest technology covers macroscopic and microscopic methods based identification by shape, color, texture, and chemical profiling with modern techniques of HPTLC, HPLC, GLC, MS, NMR, etc. Misidentification, identified collection could also lead to the introduction of toxic or otherwise unsuitable species to the market with potential health risks to end users. Two types of identification: phenotypic identification, mainly based on external morphological characters, and number two, that is molecular identification, mainly based on molecular data, DNA barcoding. Unfortunately, the phenotypic identification methods cannot identify the related species easily because, number one, these methods require expertise. Number two, even to find unique character to set keys for identification is difficult for macroscopic and microscopic features. And number three, the chemical profile or markers may vary based on geographic, seasonal, physiological, or storage condition. Development of DNA-based marker is important for authentication of medicinal plants because authentication at DNA level provides more accuracy as it will not be affected by external factors and is found same in all tissues. Using DNA barcoding to detect adulteration in different herbal plants, there are 4 steps. First, DNA extraction from different parts like dust, plant products, or leaf, or any rhizome, or any root, or bark. After that, PCR, MATK, these are the proofs. These are the markers, that is RBCL, ITS, primer. And for third stage, that is sequencing, and fourth, final stage, that is data analysis. The recent technique used for the identification of biological specimen samples is called DNA barcoding, which uses short DNA sequences from either nuclear or organelle genome. The term DNA barcode was first suggested by Paul Halbert, the University of Gulf, in 2003. What actually the definition of DNA barcoding? The DNA barcoding is a standardized approach to identifying plants, animals, and fungi sequences of DNA called DNA barcodes. Now, what are the other applications of DNA barcode? In case of taxonomies, used for proper species genus identification, delimitation of taxa, and so on. The utility of DNA barcoding. A standardized library of barcodes will enable identification of rare, native, or invasive endangered species, systematic and conservation-based studies, track ancient divergences between basal lineages. These are the important on the taxonomical and evolutionary point of view, because previously we know that the Magnoliopsida is the basal point, but now APG4 we are in the already been published. We know that we are the botanists or molecular systematics, the based on molecular systematics, and these basal lineages, now it is Amborella trichopoda, it's the basal angiosperms. So this is only possible with the help of DNA barcode. Trace organisms, evolutionary history, and systematic taxonomic relationships. Authentication of the status of our biodiversity. Adulterated herbal medicinal material establish the evolutionary links of the plant species that are missing at the moment. A DNA barcoding allows fast and correct identification of unidentified organism whose DNA barcodes have already been archived in a sequence library, such as barcode of life database, that is in short, that is bold. The premise of DNA barcoding is that by comparison with a reference library of such DNA sections or sequences, an individual sequences can be used to uniquely identify an organism to species in the same way that a supermarket scanner uses the familiar black stripe of the UPC barcode to identify an item in its stock against its reference database. Now, what are the markers used? Markers used are for DNA barcoding are called barcodes. In order to successfully characterize species based on DNA barcodes, selection of informative DNA region is crucial. A good DNA barcode should have low interspecific and high interspecific variability, possesses conserved clanking site for developing universal PCR primers for wide taxonomic application. The goal is to design primers that will detect and distinguish most or all the species in the studied group of organism, high taxonomic resolution. The length of the barcode sequence should be short enough to be used with current sampling source, DNA extraction, amplification, and sequencing methods. Ideally, one gene sequence would be used for all taxonomic groups, from viruses to plant and animals. However, no such gene region has been found yet, so different barcodes are used for different groups of organisms. In case of animal, the most widely used barcode is mitochondrial cytochrome C oxidase 1, CO1 locus. Other mitochondrial genes such as cytochrome B, 12S or 18S are also used. Mitochondrial genes are preferred over nuclear gene because of their lack of introns. their haploid mode of inheritance, and their limited recombination. Our each cell has various mitochondria up to several thousand, and each of them contains several circular DNA molecules. Mitochondria can therefore offer abundant source of DNA even when sample tissue is limited. In case of plants, however, mitochondrial genes are not appropriate for DNA barcoding because they exhibit low mutation rate. A few candidate genes have been found in the chloroplast genome, the most promising being MATK by itself or in association with other genes. Multilocus markers such as ribosomal internal transcript spacer, ITS DNA, along with MATK, RBCL, TRNS-H, and other genes have also been used for species identification. Other genes suitable for DNA barcoding are the internal transcript spacer, ITS, rRNA, often used for fungi, and Rubisco used for plants. In case of microorganisms are detected using different gene regions. The 16S ribosomal RNA gene, for example, is widely used in identification of prokaryotes, whereas the 18S ribosomal RNA gene is mostly used for detecting ribosomal eukaryotes. And in fungi, ITS, RBP1, RBP2, 18S are commonly used for proper identification. And the requirements and methods of analysis for barcoding. Molecular methods require intact DNA. It will be challenging to isolate from processed plant products because, number one, the DNA is often highly degraded, and number two, material contains high amount of polysaccharides, phenolics, and other secondary metabolites such as phenols, alkaloids, and flavonoids, which degrades DNA. For plant material, fresh tissue like leaves, flower, and buds is best, which can give 100% DNA quality. If we have extract DNA from herbarium material, leaves, flower, twigs can give good DNA. If it is sapwood, the DNA quality can be fair. But if it is hardwood, the DNA quality is poor enough. So it will be a big challenge for molecular systematics for identification. And the method, actually the total DNA barcoding methods, the first step is collection of sample. Barcoding can be done from tissue, from a target specimen, from a mixture of organism bark sample, or from DNA present in environmental sample, or we can say meta-DNA. The method of sampling, preservation, or analysis differ between different types of samples. It is recommended to collect 2 samples from one specimen, one to archive and one for the barcoding process. Sample preservation is crucial to overcome the issue of DNA degradation. The next step is DNA extraction, amplification, and sequencing. Several different DNA extraction methods exist. One extracted amplification of the extracted DNA is done. Typically, only a small fragment of the total DNA material is sequenced, typically 400 to 800 base pairs to obtain the DNA barcode. When the DNA barcode markers region has been amplified, the next step is to sequence the markers region using DNA sequencing methods. Many different sequencing platforms are available. Reference libraries and bioinformatics. Reference libraries are used for the taxonomic identification, also called annotation of sequences obtained from barcoding. These databases contain the DNA barcodes assigned to previously identified taxa. Most reference libraries do not cover all species within an organism group, and new entries are continually created. Several references databases exist depending on the organism group and the genetic marker used. First, that is bold, the Barcode of Life database, International Barcode of Life project, that is IBOL, Consortium for the Barcode of Life, CBOL, the Gene Bank, Medicinal Materials DNA Barcode database, that is MMDBD, and Genome Database for Rosacea, the GDR. There are search engine and sequence repositories, that is the Barcode of Life database. BOLD was created and is maintained by University of Gulf in Ontario. It contains single barcode library for all living species, including plant, animal, and fungus. It gives information to the researchers about how to collect, manage, and analyze DNA barcode data. The main purpose of BOLD is to provide a barcode library for all eukaryotic life over the next 20 years. International Barcode of Life project, IBOL, a group of hundreds of scientists of 25 different countries working together through IBOL to build a authentic DNA barcode reference library. Consortium for the Barcode of Life, CBOL. CBOL was established in the year 2004. CBOL contains 200 member organizations from 50 different countries and works from Secretariat Office located in the Smithsonian Institution, National Museum of Natural History in Washington, D.C. It broadcasts barcodes through workshops, networks, working groups, conferences, outreach, and training. It is a public reference library for species identifiers, which is used for the identification of unknown specimen with reference to DNA barcode of known species, which is already submitted. Number 4, that is Gene Bank. The Gene Bank is outline genetic sequence database. It is one of the most frequently used databases for genomic authentication. Gene Bank is an important storage place of genetic information. There are total 108 million entities for over 260,000 named organisms. Number 5, Medicinal Materials DNA Barcode Database. MMDB is a website that contains DNA sequences and their information and important references of medicinal records of the pharmacopoeia of the People's Republic of China, American Herbal Pharmacopoeia, and other related references. MMDBD database gives information on distinguishing medicinal materials, plant, animal, and fungi from their common substitutes and adulterates. Number 6, the GDR, that is Genome Database for Rosacea. The GDR was created in the year 2009. It contains information regarding genetic markers of the family Rosacea. Other PCR-based methods which have been applied to develop markers that help in authentication and identification of medicinal plants are RAPD, RFLP, microsatellites like ISSRS, SNPS, ARMS. And what are the advancements in sequencing technology? First of all, that is NGS, next generation sequencing, is a more advanced and newly refined version of the traditional DNA barcoding techniques. NGS is done with entire genomic DNA, whereas barcoding is done with PCR followed by Sanger's method of sequencing. What is the difference between NGS and PCR based sequencing? However, NGS is too costly and not practical as PCR amplification. The variability of NGS instrument is also limited compared to PCR because we know that PCR is very common in almost all the laboratories in India and in West Bengal. NGS requires time and skill. NGS data always needs computational skills and advanced IT infrastructure. Genome sequence is not available for all the species. In that case, still barcode is more practical than NGS. Now, finally, I'll conclude that DNA barcoding is an accurate tool for identification of medicinal plants, their substitutes, and their adulterants at genus as well as in species level. Based on literature evaluated RBCL and MATK is suitable for medicinal plant identification. Depending on the type of material analyzed, combination up to 3 genomic regions is important to provide sufficient information for identification. Because of raising demand of herbal remedies, authentication of plant material is important. Hence, it is necessary to provide an exclusive database containing DNA sequences for easy identification. Future advances of sequencing to evaluate large-scale nucleic acid sequences provide an advantage of genotyping of taxon, which will provide tool to identify species from even damaged and fragmented DNA template. From our laboratory, from the barcoding point of view, we have already done the DNA barcoding of 45 endangered and threatened medicinal plants and few liverworts from Southwest Bengal and submitted to NCBI Gene Bank, which they have already accepted and authenticated. These are few examples of our work from our team. And now, thank you to all, and I acknowledge again Professor Shivli Sarkar and all the faculty members, organizers of Botany Department, and I will be happy to take questions now. If any questions from students or any dignitaries or any scientist, please.
My heartfelt thank you, Dr. Partha Chakrabarty and Professor Amal Kumar Mondal. On the behalf of Department of Botany, Lady Bribon College, I wish to thank our principal, Professor Shivli Sarkar, for her constant support and encouragement. Thank you all the participants for your patient hearing. We are totally indebted to Mr. Arvind Prasad of AAA Tech for his technical expertise and constant help and support, without which our webinar couldn't be arranged. I request all the participants to submit their feedback in the website ladybribonbotany.in/webinar. You can collect your certificates from this website on 6th July 2020. Thank you all again and have a very good evening.